Alrighty, for the sake of time, I think we get started, but this is recorded, so obviously people will be able to watch it after this. But I'd like to welcome everybody to our 6th ever BCI Industry Academia Seminar. This is the first of 2023, so thank you all for joining. We have a great lineup of speakers today. We're going to be hearing from Professor Cynthia Chestik. She is the professor of biomedical engineering, electrical engineering, neuroscience, and robotics, a mouthful to say the least, at the University of Michigan, Ann Arbor. She currently runs the Cortical Neural Prosthetics Lab, which focuses on brain and nerve control and finger movements, as well as high-density carbon fiber electrode arrays. For anyone that knows Cynthia, you know she's a tour de force in the world of BCI, so we're very excited to have her today. And then the way this is going to run, Cynthia is going to give her talk, and then immediately after, I'll introduce Ricky, our second speaker. And then at the end of Ricky's talk, we'll have a joint Q&A for both of them. So feel free to ask questions in the chat throughout, but know that we'll have a dedicated time at the end for our speakers to answer your questions. But yeah, without further ado, I'll turn the time over to you, Cynthia.
Okay, let me go ahead and share my screen. So I assume everybody can see that. Yeah, so thank you very much for that kind introduction. I am gonna mostly talk about research today. Most of these slides are about research, but I stuck in 2 slides on career. So I think I'm gonna sort of get started before I tell you about the research my lab does. I'm gonna tell you how I came to start a lab that does these things. So I think the story starts, I went to Case Western for my undergraduate degree, which was in electrical engineering. I was there for 7 years. So you might be wondering what I was doing for all that time. I was, and Case is like a wonderful school for neural engineering, but for the first 3 or 4 years I was there, I was just a straight electrical engineer. I was super into circuits. I actually did a total of like 2 years in industry, but it was spread out across my undergraduate degree. And so it was a great way to do, these extended co-ops when I was in college. And I just did a lot of circuit design at Philips Medical Systems. That's a company that makes CT scanners. And so, it was the best way to learn about circuits is with like, a dozen circuit boards hooked together that you're constantly debugging and constantly making new ones. And so that was a sort of formative experience for me. I enjoyed every minute of that. Like, I loved it. By the time I came to the end of that, I realized that it was already getting repetitive and it had only been a few years, right? So I realized at that point that like, okay, even if I was going to stay in industry, the jobs that I wanted at Philips, for example, were the jobs that were being held by people with PhDs. So I knew I wanted to get into research. I joined a neuroscience lab, I think around like the 4th year, that I was in Cleveland. That was a Hillel Shields lab. So I started working with sea slugs, which is where, you can see the neuron, you can see the nerve, and you can see the muscle, and you can see the whole system interacting. So that was also very formative in terms of like how I think about, neurons and muscles. And so, and that, in a research lab, I was never bored. Like I never felt like, I knew exactly what I was doing. Nothing was ever easy. And I found I could not leave, right? So, and that's part of why I stuck around and got a master's degree, and realized that I kind of wanted to stay in research long-term. I did a brief stint at Guidant, which was great for, that's now part of Boston Scientific, but that was great to get some experience just before grad school in implantable devices. So that was like an internship on my drive across the country to California. So next I got my PhD at Stanford. It's a little hard to still talk about my PhD advisor, Krishna Shenoy. Many of you know he recently passed away. So I'll just say I'm pretty sure he was the greatest mentor there's ever been. So he actually, at this point, I was very into circuits. And Krishna, I had, I had taped out my first ASIC, like I really wanted to stick in circuits, and I really like neuro. But he actually sort of talked me into, how about algorithms? And, for that matter, how about monkeys, right? How about recording from a set of live neurons every time you walk into the lab? And so I joined Krishna's lab. That was an amazing experience, working on brain-machine interfaces. And then I started my own lab at Michigan in 2012, and now I'm going to be in biomedical engineering. And so, and then, like, if you go back at this point, I have, because in high school, I decided not to take AP Bio because it conflicted with choir. This entire time period, 1998 to, 2012, never had a biology class, never had a neuroscience class. My whole conceptualization of the brain is very much like those, dozens of circuit boards hooked together to make the CT scanner, right? And I think there's room for that perspective in, brain-machine interfaces, certainly, and also, I think, neuroscience. All right, so I started my own lab, and I'm actually core faculty in BME and robotics. And I work on motor neuroprosthetics. So we get to, take signals from many different parts of the nervous system and use them to generate movement commands. I think from first glance, my lab looks really broad, but it's probably not as broad as you think. So, for example, we use brain, nerves, and muscle signals because we have amazing surgeons, right? So that's all, done in collaboration. They come in and they place electrodes, so that's that. But it is sort of broad in electrical engineering, right? So we do a lot of signal processing, a lot of machine learning. We still do a lot of circuits, and I've always loved electrodes. So, it's very like broad-based EE applied to, this relatively narrow application of motor neuroprosthetics. All right, so the core of my lab is monkey brain-machine interfaces, right? So we train monkeys to make very precise finger movements. And I think that that's something that we do, differently than a lot of labs studying hand control, where monkeys actually have to acquire targets with different fingers. And then, of course, we want to interpret that brain activity in real time and decode that for controlling finger movements. And I would say, like, for students out there, the chief skill set for this, it ends up being like real-time machine learning, like debugging, things so they work in real time. Like, we don't invent new math in my lab. Like, we're using algorithms that are already always pre-existing for something else. But, getting that to work in this context and in real time is always a lot of fun. All right, so let me play you a video of one of our brain-machine interfaces. And so this is now a few years old. So on the left, I hope this isn't coming through with lags because it's nice and crisp. This is still the monkey's hand control. So he is acquiring 2 targets. Right now we're training monkeys, like, this week in the lab, we're trying to train a wrist degree of freedom to add to these finger degrees of freedom. And now this is data from a Utah array that's being interpreted real time. In this case, the algorithm is called a refit Kalman filter, and it's completely linear. So, linear regression still between brain activity and kinematics of the fingers. All of the newer things that we've been doing with algorithms are now, more and more incorporating deep learning, which has gotten much more tractable to do as part of a real-time experiment, right? And so, so we can train, iterate, retrain, like in the same day. So this is our most recent paper. It is a relatively simple neural network. Like, we were surprised by how we could get away with a network this simple. And again, it's but now this is something that we can train very quickly and tweak within experiments and things like that. So it's just a 4-layer neural network. There's a bit of cleverness on the front end. We're using convolutional time features. Maybe the only thing that's slightly odd about it is we're using tons of dropout, which is going to act as an anti-overfitting measure. But what we get from this is higher high velocities and lower low velocities, which makes sense. That linear fit just can't stretch far enough across the velocity space. So, now we can move faster and slower at the same time. And we're getting, the non-linear fit that we've always wanted now from the neural networks, without the sort of, the overfitting that might be a problem for a lot of neural network applications. All right. So I like to show in my talks at least like one brand new thing. So this is like, data that's like maybe a week old. We're now like sort of trying to figure out how to use recurrent neural networks to be useful, right, for brain-machine interfaces. And they're interesting because they can generate their own dynamics, they can generate their own trajectories. And at first it sounds like a great thing that, your recurrent neural network can just memorize all of the trajectories. But if you're trying to make something that can do continuous arbitrary control, you don't necessarily want that. So we've started trying to manage this in our recurrent neural network. So you can make them so that they have fixed points and get sort of sucked into different targets. We can train them so they don't have that. And what I'm showing you here is we actually have good signals for one of these degrees of freedom and less good signals for the other. So we have here, basically the index finger is basically acting like a classifier. It just has, a number of targets along the arc. And then the network is generating the trajectories. And then, the middle ring small finger is actually under continuous control, and those targets can appear anywhere. All right, so let me switch gears to nerve machine interfaces. So this is something where, I arrived at Michigan at exactly the right time in 2012. The plastic surgery group had developed this cool surgery for taking a nerve signal and making it big, right? So they wrap a piece of muscle around an amputated nerve. The nerve goes into the muscle, and the signals that, used to be very small are now very big. And you have, finger muscles in people's upper arms, for example. So, and I want to say again, like it's monkeys, and, we actually do a lot of the regulatory work to get the investigational device exemption to be able to do this kind of work in humans. But, it's really actually much closer together, I think, than people think. I don't think we've ever used a piece of math in human experiments that we didn't first use in a monkey or get from the literature. And so, monkeys have a lot of trials and a lot of data. And so, we've gotten a lot of mileage out of using, a lot of the same algorithms. So let me start by playing this video. My student is holding a target over there. It's an old video, but I play this one first because this is the thing that we get from the grafts alone. Most of our amputees are very transradial, so they have a lot of other musculature, but the thumb, like if you want to do intrinsic movement of the thumb, that's something where it's very, very helpful to have access to those nerve signals. But of course, we don't just implant electrodes for, in the RPNIs, we implant electrodes in, whatever muscles that person has, residually. We're controlling the hand in front, he's controlling the hand in back. And this is, so now we can classify like 10 different graphs. So that's what you're looking at here. And it's pretty quick. And I should tell you, this is one of the first times this newer participant came into the lab. So it's a calibration. We don't have to train them to be able to do these things. The algorithm here, if you're curious, is a hidden Markov model with a naive Bayes as a classifier. Our go-to is still to use a classifier with continuous control within a grass. And that's because, unfortunately, these prosthetic hands, they have a mode, right? So they have to be in, pinch mode, power grip mode, things like that. Here she's going to, fist, pinch, and point. So she'll pinch to get the Keurig cup. She does have continuous control within that pinch. So that's why she thought it was off and could just open her fingers again. Now she's going to point to plus the button. Importantly, this is all being done with parameters trained over 600 days ago. So this is very different than brain, where you do have to recalibrate the system really frequently. This is days after implant on the x-axis here. And, our parameters will maintain high accuracy, at least for these 4 grasps, across long periods of time, even in multiple postures. All right, so the last thing my lab does, and let's see how I'm doing on time, is carbon fiber electrodes. And again, this is not as unrelated as one might think, because my ulterior motive this whole time has been to use these electrodes to get better signals for prosthetic finger control, whether it's brain or nerve or whatever. So the beauty of carbon fibers is that they are smaller than the diameter of neurons, but even at those very small diameters, it's still fairly straightforward to get them, without other support, get them into the brain. And of course, they have a very minimal immune response after they're in the brain, right? So in this top image, white dots are where carbon fibers have been implanted. These green things here are neurons. If I pretend to have electrodes at different points in this control image, my neurons are only 0.6 microns away further on average in the top image than the bottom image. So, that's great from like a biocompatibility and a safety point of view. It's also just amazing for the signals, right? Like the signals that we get are very high yield per electrode. There's a lot of ways still for us to screw up the electrode, but if you have a carbon fiber in the brain, connectorized, et cetera, you get yields like this, right? And so, we very high likelihood of seeing a neuron on each of the electrodes. All right, so at this point, my lab has, over the course of, much generosity from the Brain Initiative to try to develop carbon fibers as neuroscience tools, we've distributed at this point over 1,200 devices to 48 labs. We're done, right? We have to be done. We're actually working with BlackRock Microsystems to try to sort of take some of the customers off of our hands. But what these are good for in a neuroscience, which I mean, like a silicon probe. So for example, there's a lot of people using these in nerve, in ganglia, where you can use them as intracellular electrodes. We've had people do honeybees, dragonfly, spinal cord, octopus, and certainly, we use them in cortex. And, in cortex, you're looking for the yield. And also we can process the tips of these electrodes so that they can do dopamine sensing. But at this point, I think we are done with distributing this, and we're really hoping to move this out of the lab into a company. What we're doing with the carbon fibers at this point, and I have to say, I love that there is so much industry activity now, because it kind of gives me, as the academic, a license to be one step more science fiction, which is a lot of fun. So another thing my lab does is we're trying to find simpler ways of doing the electronics. So we've published a number of papers on, how do you, reduce that front end electronics without giving up performance. There's a lot of ways to reduce it while giving up performance, but we don't want to do that. And so now we have a nice, low bit rate, low power system. So we're now trying to put a 200 by 200 micron chip on the back of a carbon fiber. So these devices are fakes. So the chip is real. We're still sort of, working out details of the hybridization and the packaging, but we're all, far enough along that we're making attempts at functional devices. And we're also, doing surgical development with, these are mechanically equivalent devices. All right, so I just have one slide left on career choices, and I am stealing with permission from Leila Hushmand. She's a PhD alum from the University of Michigan. Just gave this great career talk, and she's now in leadership at Boston Scientific. And she had this wonderful color-coded chart, and I'm not going to reproduce all of that. But basically, it comes down to, the different jobs you might be able to get, academia, industry, government, consulting, they vary along these dimensions, right? And I added one of these here, which is autonomy, but they vary in how much you can choose your topic and what that topic is. They vary in how much, whether you have a boss, they vary in what you can do when you finish them. And obviously, things like work-life balance, location, and salary matter to people. And so and what was so interesting to me about her talk was that we basically agreed on what the jobs were like, right? Like she had a wonderful color coded of like, this job is high on this and low on this. And I totally agreed, but I had a different order of importance. So this is the actual order of importance to me. And I would say this is a very professor list, right? Like, no one is dragging me away from the cutting edge. Like, I will not compromise on my topic. Like, I could not go work on pacemakers. Like, just can't happen at this point. I've been 11 years without a boss, and that's amazing. So I'm not stopping that. Older professors are pretty happy. And these things down here, I'm not saying they're not important. I'm just saying they're lower on the list for me, right? I should mention I have 2 kids. I have 2 kids and an awesome husband. So I don't think that's incompatible with being a professor. And yeah, I mean, I think everybody out there has a different list, right? And a different ordering of priorities. And I think that's, I really like this as a way to think about, which of these jobs are you going to be the happiest in? So I think I'm coming in right at about 1:20. So I'm very happy to take questions at the end, but I will turn this over. This is my lab. I'll turn this over to Ricky.
Really, really exciting stuff, Cynthia. As Yahya just said in the chat, feel free to write any questions. We'll do the Q&A at the very, very end, but definitely check out the Q&A feature and feel free to use it. But yeah, let's move on to Dr. Ricky Banerjee. Ricky received a PhD and a Master's of Electrical Engineering from the University of Minnesota. She has 12 years of industry experience from Medtronic Neuromodulation, where she has contributions as an engineer and a manager commercializing products across many different kinds of therapies, from DBS, sacral nerve modulation, and spinal cord stimulation. Ricky's current position, she joined Synchron in 2021 and is currently the Vice President of Research and Development. We are very excited to have Ricky today. So without further ado, I'll turn the time over to Ricky.
Great. Just give me one sec here. Right. See, I'm gonna hold on one sec. I'm just gonna move this to my screen. Can you guys see it still?
I see a tiny bar.
Okay, so that would be no. I'm sorry about this. Oh, stop share. Let me try that one more time. Okay, this should be better. And we'll
Perfect.
All right. I think I all right. So I have the pleasure of going second. So I had the benefit of learning from Cynthia's talk, and I thought I would add this in here. And just kind of, this is, we used to do this at Medtronic. We would call it Me in a Minute, basically, and thought I would share a little bit about myself before I jump into the presentation. So this is a little bit about me. At the top is kind of my career journey. So I started off at UW-Madison in Wisconsin. I grew up in Wisconsin and did a bachelor's in electrical engineering, and actually had some early experiences in biomedical engineering. And I don't know, just kind of decided to deep dive in electrical engineering and kind of thought of biomed as a potential future application of my work. And I think that probably worked out well because I lived in Minnesota. And if you know, Minnesota is kind of a unique ecosystem of biomedical engineering. Um, and so I did end up getting kind of drawn in that direction. I started off my career at 3M. My background is in radio waves, radio frequency design. And also I had kind of a progression from pretty early phase research through my PhD into, I would say, like an more applied technology group at 3M. So I was in corporate technology. And then really at that point, I really wanted my work to get into products. I felt like I could have a whole career where my work was kind of on the bench and didn't really get to people, and I really was hungry for that impact. So the move to Medtronic was very intentional, that I wanted my work to be in products, and it was a big jump because it was not only going into products, but it was my first experience with medical device regulation. So I actually didn't encounter that until I was at Medtronic then as an early engineer, and when I got my 50 modules of training in my like first or second week, I was like, oh, this is what it means to work in a regulated industry. Like it was kind of big shock and awe, but had amazing experiences at Medtronic Neuromodulation, and was there probably had 5 or 6 major roles, so all in neuromodulation, but contributions as an engineer. But then in program management, we have roles called core team leadership, which is about bringing together all facets of bringing a product to life. I had experiences from early phase design, translation of technology, all the way into what does it mean to launch a product and then sustain it in the field. So there's fantastic experiences, which is what led me to the position at Synchron as VP of R&D. And I try to describe what those experiences are like, and kind of just came up with this word cloud because it kind of ended up being that, with all the different experiences that I had from all the different groups and phases. So Synchron, I'll talk about Synchron and tell the story of Synchron. Why did I switch from a big company of Medtronic to Synchron? I was really drawn to this idea of like major technology translation. I had the opportunity at Medtronic to do that and contribute in smaller ways, I guess I'd say. And so this was kind of like a really, really big, impactful thing. The brain sensing technology was actually very connected to what I was doing. So Medtronic neuromodulation as the deep brain stimulator with the sensing and for tremor disorders, and so I knew something about that. So I had some kind of connection that I was like, okay, yeah, this is like really connected to the space I've already kind of found a love for. And then I just found Synchron had some really engaging people, and people were drawn into that. So I was like excited about meeting lots of different people and bringing together people from different industries. I won't go through the rest of what's on the slide, but it's kind of supposed to be me in a minute. So it's not just my career journey, but a little bit about me as well. All right. So Synchron. So you may be aware Synchron is doing the endovascular brain-computer interface for severe paralysis. And I'll share a little bit about the story of the first in human experience. In about a year ago, Tom Oxley, who's the CEO and one of the co-founders of Synchron, gave his Twitter account to to a man named Phil. And Phil was able to tweet out using his BCI, "Hello world, short tweet, monumental progress. No need for keystrokes or voices. I created this tweet just by thinking it. My hope is that I'm paving the way to tweet through thoughts, Phil." And so with that, I'll introduce you to Phil and Rodney. They're two of our patients or our subjects in Australia. And this system was implanted to help these patients with ALS. And so for Phil and Rodney, their speech was becoming garbled, their hands, they were becoming more and more paralyzed. And so this device really helped them regain that ability to communicate. You can see the brain waves across the screen, and you can see how slowly they're typing, and it's fairly slow. But this is really kind of those first days, right? The days of that dial-up modem when in the early days of the internet, that that some of us remember and some of us have heard about. This is Phil and Rodney on a group chat with Tom, and they're teasing him a little bit. Some of you may have seen the TED Talks that Tom did, and they're teasing him a little bit about the big stage that he's going to be on. And that's Tom responding, "I've got this, fellas." So there's over 5 million people living in the US with paralysis, and it might be due to different conditions. So it could be stroke, could be multiple sclerosis, demyelination disorders, neurodegenerative disorders such as ALS or muscular dystrophy, trauma, or neuromuscular disorders. And as I mentioned, I'm an employee of Synchron. All of the devices I'm going to describe are under investigational devices. I think Cynthia mentioned the Utah array. This is a close-up picture of a Utah array. This device is very foundational and seminal to research in BCI, and continues to be used a lot. I think that Tom Oxley was studying as an endovascular or neurointerventionalist, and he was reading about BCI, and he actually had the opportunity to talk to Lee Hochberg, and it was really inspiring to him. And he started to think about the domain that he was working in. And if you're putting devices already in the blood vessels of the brain, what if you could create devices that could extract these electrical signals and do sensing? Could you not get to all the different corners of the brain at some point, especially if you're piggybacking on this industry? It's already trying to navigate that for other applications, such as stenting for strokes or conditions like that. This is kind of showing an image of that kind of a stent retrieving a blood clot. So some of you may know, I don't think I even realized that this kind of neurointerventional endovascular approach, I mean, obviously it's piggybacking off the cardiovascular and peripheral vascular world, but the neurovascular space is really still quite new. Like it's really kind of in the last 10, 12 years that the first FDA approvals of these kind of devices came about. But now we think about this as very standard of care. There's angio suites everywhere. People are looking at how do you reduce the time of someone coming in and retrieving a blood clot. And there's so much innovation in catheter and stenting technology, and how do you get to all the corners of the blood vessels in the brain. So it's piggybacking off of that. The founding team members initially approached Medtronic Neurovascular, and they took a couple of stent-like devices and did an animal study. That publication was published in Nature in 2016. After that point, then they started developing a system for human applications, and for chronic implants for human applications, typically you look for a system that's kind of off the shelf, so it's safe. You're looking to establish foundational safety, and then you build upon that to create software, a research system that allows you to explore and investigate. So that first system really enabled that. The device in the chest, it's implanted in the chest. We call that the IRTU, and then there's a lead body that connects to it, and it goes up through the blood vessel, and then the stentrode itself is deployed at the top of the head in the superior sagittal sinus. This is also where your motor function is, and so it's a great location to place that device, because then you can capture motor function. And then on the outside, the device is powered with this external device on the skin surface, and then those signals are captured and processed in the signal control unit, which is then plugged into the laptop for the outcome. Prior to the implant, the patients have to undergo some extensive screening, ensuring that the anatomy and the vasculature that they have is appropriate for the device. We also have been using fMRI to evaluate brain function. You want to make sure that the functional circuitry is still there for motor intention and motor action, so that that's what you could capture. The surgical procedure itself, you gain arterial access for angiograms, and then jugular venous access is how the stentrode itself is deployed. The stentrode is inserted into the superior sagittal sinus and deployed there, and then there's a surgery in the chest for the pocket for the IRTU. There's a tunneling of the lead that goes between the stentrode and the IRTU, and so the whole thing then is subcutaneous. This image here is showing how what it looks like after the stentrode is deployed. So you could kind of see a slight distending at the top of the head. You can see that lead body coming down the big major blood vessel there. And then this is showing under imaging what the stentrode looks like when it's in place. This is an overlay of that pre-implant MRV, the x-rays of the electrodes, the functional MRI, and the anatomy that's pre-implant. When you overlay them, you can start to get a picture of where are those electrodes and what's their position with respect to activation in the brain. This study was completed in Australia. It was 4 patients and then with 12-month study and follow-up. So the final results of this were published in JAMA. There was a recent article actually of the full study with 0 serious adverse events. Also kind of key outcomes we're looking for. So from a safety aspect, you look for device migration. So that's kind of one of the things is making sure that the radial force is sufficient such that it stays in place. And then looking at how an endothelialization occurs. And endothelialization actually helps improve the signal quality or stabilize the signal quality. So it's actually been a benefit to the signal quality overall. The first use of system has been, a key metric often published for BCI systems. And you could see how we improved greatly. So with the first patient, there was a lot of learning in terms of how to help that patient use that system successfully the first time. And so then you could see that was applied very well across the other patients. A number of you are familiar with looking at the different motions and signal processing techniques to evaluate if you can see a difference. So from patient 4, you can see that you can see a difference when the patient is at rest versus a left ankle versus a right ankle, left hand, etc. Of course, or you could see those differences, the more you can extract and get repeatable results. So what is the patient doing? The patient is thinking about moving their foot, tapping their fingers, waving their hand. It's a motor intention signal that can then be brought forward and used in this way that we call a switch. So what you're seeing on the right, when you saw that animation, was an interface that's common on all of our devices. So these type of accessibility user interfaces exist for other needs. And if you can connect this BCI system with that ecosystem of accessibility platforms on devices that we have, then you're kind of also piggybacking on a wide array of devices and tools that are greatly accessible for this patient population. This is showing some amount of signal conditioning. So if you're in the signal processing domain, you're very familiar with how much filtering you have to do sometimes to see the signal and how much cleaning and post-processing of the signal you have to do. So just kind of taking someone through what that might look like. So some of you might be familiar with this dinosaur that jumps over the cactus. We connected our synchron system up to it, and you could see this little bar here showing the patient as they're thinking they have to jump over the cactus by getting it over the purple bar. This one is showing the patient sending a "What's up" message. I think one of the things that's interesting here is you'll see that the patient has the ability to do kind of a long click. So right there, this long click is allowing the patient to be able to zoom in and select something. A lot of these systems, this one is used in in tandem with eye gaze. So a lot of these patients might already be using an eye gaze system. So one of the things we're assessing as we look towards commercialization is how do we enable an ecosystem for these patients? They already maybe have unique systems, unique tools that they use. You don't want to take that away. So how do you enhance that experience for them such that they can keep using tools that they have that are working well for them? Then this one's kind of nice because it reminds us that nowadays when we're using our computer, it's not just a computer, it's like the world is connected. So when we talk about the Internet of Things, we may have kind of thought, oh, why do I need a smart light like that? But you kind of marry that with the idea of someone who needs an assistive technology and realize how wonderful that is that he can now use his computer to turn on a light in his home and control his environment. And then typing, of course, is metrics of correct characters per minute, accuracy per word. So kind of in the realm, I think, of BCI technologies. So where are we going now? So with the success of the 4 patients in Australia, we built a team in the US and we'll continue to build that team in the US. We're based out of New York. And we started the command trial. So we have FDA ID approval to do this command trial, which is our safety study in the US. So we've implanted a number of patients already. We have a few more to go. We're working with Mount Sinai Hospital and University of Pittsburgh Medical Center to complete that study. So that's kind of what's happening right now. If you know people that might be interested, these are some key contacts, or if you contacted me, I could forward your information. This is a nice photograph of one of our patients in Australia and his wife, and it's just been a blessing and kind of a new lease on life for some of these people as they're able to communicate more. And I think it was this gentleman that said, it gives me great comfort to know that I'll always be able to tell my wife that I love her. So it's just been really an amazing technology. I'll open it to questions.
Great. Thank you so much, Ricky. We'll now use the rest of the time to do a joint Q&A. You can ask questions related to career, related to the technical material that was presented. So feel free to use the Q&A. I'll start with a general question for both of you. Cynthia, obviously with running a lab, and Ricky with the multiple research teams you've run, what is sort of the wisdom that you've probably given now to many people many times that are at sort of a crux between choosing a career in academia, choosing a career in industry? What sort of advice do you give people to what drives them? You both hit on this and you both went through this personally, but what do you tell people? And feel free, whoever wants to take it.
I'll jump in. I have a personality test, but I think it's more for research versus sort of, rank and file engineering. I say, okay, if you like feeling good at your job, go be an industry engineer. If you are easily bored, go be a researcher. Right. So, because, researchers never feel good at their job, but if you feel good at your job, there's a very good chance that you're doing something that you're going to be doing for a very long time. So.
That makes sense. Ricky, do you have anything to add?
Yeah, I think when I was at that moment, I think at every kind of crossroads, I like to talk to people who kind of gone down both paths, or get an experience that's down both paths. So at that point, maybe a year from graduating from my PhD, I actually, with intention, took, we had a class called New Product Design and Business Development, which was about bringing together business students and engineering students to do a project and taking you through both the business side, developing a business case and doing a prototype. So that was kind of like my toe in the water on the industry side. And then I did some preparing future faculty, what would that be like? And and talking to people on both sides. And I think for me at that moment, I was like, boy, like I don't feel ready to be a professor. How would I guide students? I don't know what industry is about. I mean, I don't know. I think that's probably a true statement that if you've gone down the academic path, you don't know what industry is about. But at that point, I was like, I don't know, how would I guide someone? And so I, decided to go into industry. But then for a while, like I taught, um, you know, a section or something still at the University of Minnesota. So I guess I had the ability to stay connected to the University of Minnesota. I stayed more connected to conferences and professors and papers, like a little bit more in research. And so it was kind of a process, I think, for me of, and then at some point I was like, wow, industries are really good fit for me. I like taking deep technical things, connecting with deep technical people, but being able to explain them to a broad, cross-functional, cross-disciplinary audience. So that was a really great spot for me, because a lot of what's in industry is you have to be really good at like communicating to this breadth of people to connect.
That makes sense. And I guess before we get into some other questions, while we're talking about this space, what is it that you both look for applicants looking to join your individual research teams? Like, what do you look for in an applicant, especially if they've done a PhD, or let's say they haven't done a PhD? What sort of is in your inclusion criteria?
Well, I think that, okay, I think that the hardest thing about PhDs and research is not actually the technical side. Like that's really important, that's something that you've got to have. But the thing that makes it super challenging is the independence, going open loop for long periods. And if somebody's going to have difficulties in the PhD, it's going to be about time management, right? So it's, it's the dizziness of freedom, which just gets worse and worse and worse in academia, or, better and better and better if you enjoy, providing all of your own structure and, deciding what to do and managing your time.
So I guess I hire people kind of at all levels, interns, PhD postdoc, experienced people. And when I'm hiring for a PhD, typically I'm probably have identified a unique, deep skill set that I'm interested in. So I usually ask a question about, take me through a challenging problem that you had and how you navigated it and how you resolved it. And I'm looking for pretty deep understanding of root cause and problem solving, and kind of being taking me through an understand that they understood what was going on and how did they manage it. So I think that's kind of a key aspect. And then I'm also kind of interested in like what are their motivations. So commercializing medical device is challenging, and what helps you persevere through the challenges is really being grounded in a mission, that you really believe that what you're trying to do is going to benefit people and benefit a big population of people, and you're very motivated by that. And it helps on the day-to-day frustrations or challenges that you have, to kind of have that as your North Star and your compass. And so if that's not aligned with your motivations, then I worry that people are going to hit their bumps in the road and not have that wherewithal to persevere, that they'll get frustrated and not seek to understand why the situation is that it is.
Yeah, I see what you mean. On this topic, there's a question in chat related to skills. So Will is asking, what skills would you recommend to hone before applying to pursue a PhD in BCI research? And I guess, Cynthia, where this is closer to what you deal with more, yeah,
I think, the skill sets I sort of check on, you don't have to have done it all at the undergraduate level. But, questions I ask in interviews is, I kind of want to make sure they have circuits background, linear algebra background, and stats background, right? A lot of the machine learning courses, start at the graduate level. So it's okay if they haven't gotten into some of that. But I think those are sort of the three things. It's very hard to start a PhD doing neuroprosthetics without circuits, linear algebra, stats.
Makes sense. We've got some technical questions related to Synchron. So the first one says, will the Stentrode system need to be tested for MRI safety compatibility?
Yeah, that's a great question. So I actually led the MRI compatibility team at Medtronic for a while. And the current system that's in Australia, the research system is contraindicated for MRI, basically. And so, I mean, I think generally, we look to our physicians to understand what does this patient population need and what would be beneficial to them. And so, if we are hearing that that MRI access is important for this patient population, then I think we would look to seek for MR conditional labeling. So, yeah, it would have to be evaluated.
And a follow-up question to that, will the Stentrode be used for treating epilepsy?
So it's currently not in our immediate plans. I think I get this, I'll say, type of question a lot. I think a lot of people see the Stentrode and then they start to imagine, they're like, where else could you put it? What else could it do? And so it is kind of a wonderful device in that way, and that it kind of proliferates this idea of where could the technology go. But I think since we're in a pathway of trying to commercialize, get into a pivotal study, we kind of have to stay focused and the application we're going after is paralysis.
And I have a technical question for Cynthia. So for those who have done work in amputation, the RPNI approach is definitely a game changer, to say the least. And you talked a little bit about sort of the amount of training data that's going into training the decoder. Is there anything that you've gained from what you're doing in monkeys and how you're in the networks you're setting up there that you could potentially leverage to create more of a co-adaptation system where you can speed up the training process?
I mean, I think I have to say we have so much less data from our human subjects, just because it's really boring to do the same thing 80 times in a row, even if you try to just get the same amount of training data. So we are eyeing our monkeys who do 2,000 trials in a day, in thinking about how to adapt, particularly the deep learning approaches to RPNI. So we barely started that. Honestly, it's a lot of the work was figuring out how to move from MATLAB to Python for real-time experiments. That is a giant pain that every BMI lab went through at about the same time. And so we finished that for monkeys. We actually started it immediately then for the RPNI subjects, but we haven't gotten as far as really doing something that we're going to use on a day-to-day basis with our patients. So we've done some starter feeler algorithms, but I absolutely am eyeing the monkeys with who actually have implanted EMG. To try to augment our data sets for the human studies.
That makes sense. And on a similar note, how much of a bottleneck do you think hardware will be in this space? I mean, you mentioned with most of these prosthetic hands being largely mode-based between close, open, pinch. How do you solve a hardware problem where you're gaining so much from what's going on in the data space right now?
So I actually think, for the nerve and the RPNI application, it's not that bad. I think that, any of a number of existing devices could work. We're trying to figure out, what's the best way to make that happen, right? Because everything I showed you was 8 bipolar channels or 12 bipolar channels. If we had 32 bipolar channels, it would be amazing. There's still a lot of, room in the arm. Those electrodes are extremely small. I think the brain application is different, right? I think that, I'm after able-bodied hand control, right? More degrees of freedom, things like that. So, I think I need more signal. As a start, I'd love to have 100 actually good electrodes at all times, right? So high yield electrodes. And, what captivates me about the carbon fibers is the possibility of one day having 1,000, right? And hoping that the safety profile is such that you could eventually build a 1,000 channel neural interface and really start to restore function that would be very hard with fewer signal sources.
Yeah. A question in chat for Ricky. Hey, Ricky, how do undergrads, new grads reach out about interning at Synchron? What sort of experience do you look for at that level? And on a similar note, maybe you could also speak to any hiring at Synchron, sort of what positions people are looking for, all that stuff.
Sure. So we do have a website, I think it's synchron.com, that has hiring posts on it, so you could definitely check that out. At the intern level, there's various skills that we look for, kind of depend on which team is hiring the intern. Generally at a startup, I would look for someone more senior. So sorry, I have a little bit of a cold. And drink some water.
No, you're fine. But is it by more senior, do you mean PhD level and above?
I would say at least upperclassmen and above. I think when I was at Medtronic, we were doing more like freshmen. Sometimes we would do freshman, sophomore, even or like late high school sometimes. So I just like, I think at minimum some basic coding, basic bench skills are really helpful just to have a meaningful project. I'm not going to say that's, so I would say in my team generally, I think those are the types of things I look for is, and BME programs can be different. So like, as I look, I don't know, like for example, like tissue engineering or something might not be as aligned with the types of skills that I'd be looking for, which is more like, you know, neural engineering with that kind of electrical underpinnings under it. So sorry.
I see. And for the person who asked the question, are Synchron internship applications open? Do you know?
You know, I don't know if we have a post right now. How do people reach out about interning at Synchron? I don't know if I have a great response for that. I mean, we hire very few interns because we're a startup. So I mean, the best way to reach me is kind of through faculty. So maybe contact Hunter.
wait, don't give that advice. We'll go to the question that just came into chat. So great talk, Cynthia and Ricky. This is from Davida Valeriani. Do you think implanted invasive BCIs will also be used in the future for able-bodied people? Or do you see them as a useful tool for rehabilitation restoration?
So the way I answer this question is not in my lifetime. Also, nobody's working on it. Like whatever sort of, you know, gets sort of said, I think realistically, no one's actually working on that right now. You know, if you think humanity has, you know, a million-year future, like, you know, you never say never, right? You know, maybe even longer. Like, so, you know, you never say never, but not in my lifetime, not anytime soon. And basically no one's working on it.
Great answer. Do you have any addition, Ricky?
Yeah, I think right. I think maybe coming from this domain or understanding the depths of it, you're like, first of all, this is a very challenging field. Like it's not straightforward to read brain signals and create function. And then second of all, and you know, doing something that has this kind of like risk, like taking on a surgery that even a surgery that's less invasive through the vasculature, taking on that kind of risk for something that doesn't have medical benefit, yeah, just kind of seems like a tall order. And then for something that has, you know, functionality, but yeah, I think you're kind of in the sci-fi when you're down that pathway of really robust utility.
Makes sense. Alrighty, we are coming up on time, so I think we will end it there. I want to say to everyone who attended, thank you for attending. This will obviously be posted online, similar to the last ones. And then for our speakers, thank you both so much for taking the time today. We are very lucky to have you both. So thank you again.